end of one unit coming up, starting another. So we're ending the genetics, just had a couple of little animations to go through. There's not a lot more. A little commentary on that, and then followed closely thereafter by the start of viruses, which is, I think, the most extensive amount for the next exam. So I do want to get a start today, at least give you the basics on that. And again, I flip flop probably between the two PowerPoints that are going on. So we will get back to where we were, and it's fairly straightforward. This is the idea. This is the most common of all of the transfers. This is the one that happens so rapidly, so frequently, so alarmingly, if you want to think of it that way, particularly with the spread of resistance. And it's called conjugation. It is a contact transfer. The other ones don't require direct contact. This one does. Okay. So that's interesting. And though it's not easy to see this particular appendage here, this is the so-called fertility or F pilus. And what it is a hollow tubule that's distinctly designed for genetic transfer, and not all organisms have it. And while I'm interested in your favorite animal, color, movie, food, or song, for those out there not seeing this in real time, so I'm going to put this up. It's like secret questions on you have to reset your password. What's your favorite movie or genre? So, the F plasmid. So, and we say if it has a plasmid, we call it F positive. So the F plasmid really has two features that require its identification. One, it's the ability to make the pilus, the F pilus, and two, the ability or whatever the genetic ability to accomplish the transfer. That's what an F pilus has. So those are to accomplish the transfer of the genetic material, or if you want to just put it very simply, to conjugate. It can have other stuff. It can have resistance on it. In that case, it's also an R plasmid. The F plasmid is very specific. Most bacteria have a fair amount of plasmids because, again, they're useful but non-essential pieces of genetic material that play an important role. So, and you can transfer more of them at one time, and the transfer doesn't take terribly long, so it's limited by time, the amount of time that these bacteria are stuck together. But the unique part about that, when the F plasmid is transferred, the recipient cell now becomes F positive. And it's almost, you're going to see a very unique mechanism called the rolling circle mechanism that allows the one cell that's donating the plasmid and whatever other genetic material to remain F positive, even though it's in the process of transferring that genetic material to yet another cell, to another bacteria. So that's pretty unusual in and of itself. And we utilize this technology extensively in genetic material. And I'll just, you don't have to know it, but I'll show you the other animation, which is called chromosomal transfer or chromosomal DNA. And that's really the framework of genetic engineering in the way that we can genetically modify organisms, and you see them everywhere, whether it's pharmaceutical agents or whether it's foods the technology has been around for a while. Now we see more of a buzz from it, and unfortunately, with all things, there's everybody has an agenda about whether they're good for you, bad for you, or useful, whatever. But I try not to get into that because that's in a different category of university studies than biology, I think. Okay. So, nevertheless, so you can call them a conjugative plasmid. That's fine. I don't get into the nomenclature. But to me, it's about the F plasmid or so-called fertility plasmid. We certainly, E. coli is used a lot because we understand it, and it has some very unique forms. That's what you'll see in the next animation that is there. And a lot of times you'll see F positive, meaning they have the plasmid, versus F negative, etc. So I'm going to flip this over, and I already cooked up the animation, and you should all be able to hear it. I hope. Did I volume.
Conjugation is a mechanism of gene transfer that requires direct contact between donor and recipient cells. F plus cells carry the F plasmid, enabling it to make an F pilus and act as a genetic donor. Recipient cells that lack an F plasmid are referred to as F minus. The F plasmid encodes for the F pilus, a protein appendage that attaches the donor to the recipient cell. The first step in plasmid transfer is contact between the donor and the recipient. The F pilus of the donor cell recognizes and binds to specific receptor sites on the cell wall of the recipient cell. The plasmid then becomes mobilized for transfer when a plasmid-encoded endonuclease cleaves one strand of the plasmid at a specific nucleotide sequence called the origin of transfer. A single strand of the F plasmid, beginning at the origin of transfer, enters the F minus cell. A complementary strand to the single-stranded plasmid remaining in the donor is synthesized via the rolling circle mechanism. Once inside the recipient cell, a complementary strand to the single-stranded DNA is synthesized. When F plus and F minus cells are mixed together, eventually all of the cells become F plus.
Okay. Oh, hold on. My stepson is taking care of his grandma this morning, so I'm getting buzzed. So I told him I'm in class. But nevertheless, let me get back here. Sorry. Here we go. So I'll just go through the salient features that are associated with it. So this happens with it because there's so many bacteria, they bump into each other. And the unique thing is that when it occurs to one, and then the recipient cell becomes F positive, not only does it get all the characteristics from the plasmid, and it becomes F positive, it replicates as F positive, and this is how it does it overnight, and then it conjugates with other F negative cells. So it doesn't take long when you think of how rapidly, if you remember the growth curve, how rapidly they replicate. Now they're continuously making more F positives. It's also making F positives that will replicate as F positives that eventually all of the cells become F positive in the population. And it's all based on this. Now, what's interesting about these chromosomes is they're separate and distinct from the bacterial chromosome. And it's a little bit easier transfer for this to accomplish in a sense of, let's say, transferring resistance, like for MRSA, for methicillin resistant, making a population become MRSA overnight. It's not that useful for genetic engineering when we're trying to modify the organism to just make something very, very specific, typically a byproduct. Pardon me. So what happens here, they bind to a receptor. They have to have a receptor, and it opens this passageway. The passageway, and obviously you can tell by the depiction there, is larger than you think. So more than one plasmid can be transferred at a time. So that we're only showing you one in the animation, but more than one, easy. So as we look closer at it, this plays a role in genetic engineering, the origin of transfer. We want to make sure that the genetic material is close to that, as you'll see in the next animation, though that's not something I'm going to ask you questions about. So at the origin of transfer, we know where it starts. And this rolling circle mechanism is simultaneously the one of the double-stranded DNA, one piece of the basically identical DNA strands is going to the recipient cell, and it's being simultaneously replicated. So if this cell was magically, if this plasmid in part started to disappear, that cell would become F negative because it wouldn't have a complete plasmid. So what happens here is that it permits the simultaneous replication, so the F positive stays F positive. And once this, and it takes a couple of minutes for this circle to be completed, to transfer to the other side and then rapidly replicated, once it's completely replicated, it ends. Now this cell that was the recipient cell is F positive. So whatever has gone in there has gone in there. And it stops, they separate. Now you have two F positives that are there. And so the transfer ends. But again, these cells will replicate in that regard, in that fashion. So it's a little bit different. Want to do that? It's a computer. So it's a little bit different than this transfer of chromosomes. This is the genetic
Sometimes
engineering of
the
it.
F plasmid becomes integrated into the host cell genome. The host cell is now referred to as HFR, which stands for high frequency of recombination. The HFR is able to transfer some of the host genes to the recipient. The sex pilus contacts the recipient F- cell and pulls the cells together. The donor chromosome is transferred as single-stranded DNA starting at the origin of transfer. Gene 1, which is closest to the origin, is transferred first. Segments of the integrated plasmid are at the beginning and the end of the DNA being transferred. It is theoretically possible for the complete genome and the F plasmid to be transferred to the recipient cell. However, this does not happen because the donor and recipient cells will separate prior to the complete transfer of the donor chromosome. The transferred DNA becomes double-stranded. The donor DNA is integrated into the recipient cell's DNA by homologous recombination. The recipient now carries transferred genes but remains F-, whereas the donor cell remains HFR.
So the distinction here between these two events is very straightforward, very simple. It In this particular instance, it's a unique kind of cell. This is the E. coli we use, this HFR E. coli, because it willingly incorporates genetic material into it. So the genetic engineering part and actually using transformation is part of the process to generate these plasmids. That's how the two processes work together. What they do is they sort of incorporate the genetic material close to this area. The neat thing about this has to do with something that bacteria produce that are part of the mismatch repair. They're called restriction enzymes. Restriction enzymes are fascinating because we know they look for certain what are called palindromes, these little repeat segments of DNA. So it'll be like ATTA, CCCGGG, and there are certain areas where they're on either side, and it creates an area where the restriction enzyme will chop it, and then you can create what are called, they have a fancy name, they call it sticky ends, basically mated pieces of DNA in the laboratory that they know a certain amount will adhere to that DNA. So it's a rather elaborate process of genetic engineering, but the way they're able to do that with the particular enzymes is to put them here. So they put them close to the origin of transfer, realizing that you're just not going to have enough time where they're going to stay conjugated for the whole thing to transfer just a little piece. Like I said, I used to ask students questions about this. It's just too complicated in a sense of it confuses with the one that we really need to be more familiar with, which is the conjugation, the simple conjugation and transfer of the F plasmid, which frequently transferred bacterial resistance. So here, this is more, in a sense, if you were doing a graduate level and studying to do genetic engineering in the lab, this would be more apropos of something to look at. So effectively, all it does is it transfers a limited genetic fragment because these cells will suck up any DNA that comes their way into the nucleoid. Bam, it does that. Now mismatch repair and all that type of stuff goes on, but effectively you've altered the cell genetically. So to me, that's kind of the interesting area that's associated with that. So they show you these from a PowerPoint perspective. They show you these, and again, I don't spend a lot of time going over it per se. This is really the same thing that you see. Here's the HFR cells. Theoretically, it's reversible. It really isn't. If it doesn't work, it doesn't work. Then you just discard that. The E. coli that hasn't worked the way it's supposed to. So theoretically, it's not necessarily a one-time event, but in the reality is in the laboratory it is. That's there. So you can go through all of these different things that are part of it. And collectively, all of these fragments that move have a name that you probably should remember. They're called the mobile gene pool. So much of the variation in gene pool of a single species is from, so really, if you look at E. coli, less than half of the E. coli genes are found in all strains. That's the core genome. That's kind of what makes E. coli E. coli. Everything else has come from plasmids, and we're going to look at transposons and other fragments of DNA called genetic islands, genomic islands, or DNA from viruses, phage DNA. So it's a relatively limited amount that's there. And so double-stranded DNA with an origin of replication, that's sort of what plasmids are common, non-essential, but allow survival. Typically just a handful of genes that are there, but there can be more. Relatively narrow host range. And again, range applies to what it can contact and transfer the material to. Okay. And then mobile plasmid requires conjugative plasmid, F plasmid, to be able to move along and do all those types of things. And so you can see antibiotic resistance, antibiotic synthesis, increased virulence. So Yersinia plague, Shigella dysentery, toxins, oil spill degradation we use for Pseudomonas, anthrax toxins. So there's a lot of different things theoretically and in real life that are there. And I'm not going through it. I just want to show you this one about here's resistance plasmids. They can have a variety of resistance. They're not limited to one type of antibiotic that they are resistant to. They are the big problem. That's the hardest chore that you have. You're happy when something, if you're a clinician and you have an antibiotic that's a good choice and it's going to work and the person's not allergic to it, it's like, it's a wonderful day. You got something that's going to work. You anticipate. And hopefully they don't have a problem with side effects or things like that to it. There's a whole, and we'll get into that a bit with antibiotics. And here, the various kinds of insertion sequence. This is meant by these, these are somewhat, they'll look for these different repeat segments that have these. I love the word palindrome. It's like race car. You turn around both. Otto, Bob. It doesn't matter whether it's forward or backward. It's a name that works both ways. I love that. Have fun. You're awful. Why do I get excited about these things? Now, here's one. Transposons. This is the classic example. If you think MRSA is bad, for years we had one drug that was a drug of last resort for staphylococcal infections. It clobbers gram positives. It doesn't even work. They used to use it orally, and this is why they stopped. Okay, it's called vancomycin. Okay, so patients was infected with Staph aureus that was susceptible, as nearly all of them were. Okay, so normal microbiota included a vancomycin-resistant strain of Enterococcus. Enterococcus faecalis is a cousin of Streptococcus. It used to be called Strep faecalis. It's a gut bacteria. And what happened was, eventually, when we used to use vanco excessively orally, inappropriately, eventually strains developed with resistance, and it became on a plasmid effectively that, and then a transposon, as you will see, latched onto it I'll show you in the animation that's there. So it grabbed on the Enterococcus faecalis plasmid, was transferred by conjugation in this point, but the transposon made it into a variety of bacteria. And so what happened, even though the plasmid was destroyed, the vancomycin resistance was incorporated. And then we began to get these superbugs called Visa and Versa. Vancomycin resistant Staphylococcus aureus and what's called vancomycin intermediate. You didn't do the antibody, you didn't do the Kirby-Bauer test in lab with the little discs yet. Coming. We talk about that in the laboratory perspective, things like that. Voila. Different things about bases, ability to cause disease, pathogenicity islands. This is the restriction modification system. So restriction enzyme cuts the DNA in specific located. And what happened, it's like a little race. Restriction enzymes are chopping up DNA. Modification enzymes are methylating. They're putting a methyl group, an organic group, on the DNA to prevent the restriction enzyme. So they have a little race. And that's kind of what mismatch repair is about, if you want to think of it very simplistically. We even order these here We don't have a giant research university, but we've had a number of students doing research, ordering these CRISPR systems. Okay, small segments of phage DNA that recognize specific DNA if it invades the cell again, and so you can order different ones that are there to be utilized in research. And college lab like ours doesn't do it with pathogens. They typically do a lot of studies with yeast or E. coli, with bacteria that are unlikely to be problematic, without getting into all the different arrays that are there. I'll show you the transposon one, and then we'll be done with this. So transposon shifting, there's two of them. I got to find the right one. So maybe it's simple transposition. We'll see.
The most
No.
direct mechanism for movement of
Yeah. I had a chance, 50-50. Shifting segments. This one I think shows it a little bit better.
Transposons are segments of DNA that are capable of shifting from one location to another. A transposon enters the cell by being carried on a plasmid. A transposon can then move from the plasmid into the host cell genome. A transposon can move from one site on the host genome to another site on the host genome. When some transposons move, they replicate, leaving a copy in the original position. A transposon can also move from the host genome to a plasmid.
And that, if you remember the very start of this chapter with Barbara McClintock's research about the corn and those patterns, she could only explain it by a phenomenon like this. And even though they had doubters, eventually it was proven to be correct. So it's very interesting. And and again, that's going to be sort of the hidden subtext about antibiotics, is all of these mutation and genetic reassortments because of the relative genetic lability, meaning there's a certain degree of instability where gene segments can move, and fragments of bacterial genetics can move, and phage viruses can carry it. So it's really, to me, it's a fascinating area that is, for whatever reason, students at your level, it seems to be one of the more difficult units. So I've tried to make the exam questions a little more simplistic, and I generally have like between 10 and 15 on the next exam that are there. I think the bulk of it comes from like maybe 30 questions from viruses and 20 questions from the chemical and physical methods control, and about 15 from this, just off the top of my head. Subject to change without notice. God, awful. I'm trying to have fun, and you guys are like a pain. Next chapter. Thank goodness we're doing the next chapter. So this is the end of that. This is the start of this. I cooked up her Cowan's chapters first before I looked at the Nestor chapter, which I use just a couple of slides in all the time. And this is the idea. These are the acellular forms. So we took a little step back from doing strictly the anatomy, like bacteria or eukaryotic microbes, and then we dealt with the physiology of it. We dealt with how they grow. We dealt with how we get rid of them. We dealt with how they genetically reassort, if you want to call it that. Now we're looking at things that are not really living. Again, you'll see the term, and I'll use it. How do we kill a virus or kill a prion? Well, you theoretically can't, because they're not alive, because they're not made out of cells. So collectively, they're referred to as acellular forms. The prions we'll look at the end, the viruses first. Okay? And so better terms for viruses than alive or dead. Great. Viruses, it's a question on the exam. Viruses can infect all living things. So the bacteria can get a virus just like you and I can get a virus that are there. Okay. And it's a good unit and this should be a unit to frame it that you guys should be very interested in. This is where you learn about what really COVID does. And whether it is severe as everybody made it out to be, or exactly what the origins were of COVID, which I still do not think are terribly clear. The reality is it changed our lives from this time forward. Like my wife and I are we're looking at booking a trip. What happens if there's a COVID outbreak? You know, does your trip insurance cover it? If you're going to another country, what are the restrictions? If you travel to certain countries, and I found this out yesterday morning, people used to buy trip insurance, and you were hoping that if you got lucky, like somebody got sick, that you get. A lot of times they didn't pay. It was like buying those extended plans on your car. They only covered limited things, at least in my experience. Maybe you had a good one. And all of a sudden, if we want to go, like we were looking at a trip to Israel, if you want to go to Israel, you have to have trip insurance that includes medical insurance to cover you in case you get COVID to be treated there. So I said, but we both have Medicare. Oh, too bad. The lady was very nice. She didn't go, you're SOL about that. See, Sadie got that. Because it's inappropriate to say shit out of luck in class. Oh, never mind. So understand it changed the world. And now everybody is flipping out about things like that. I can't tell you how often my colleagues will come now, norovirus in somebody's school. We're going to talk about RSV. Everybody magically forgot about influenza, which is the single largest killer of all time at one time, called the Spanish flu back in 1918 and 1919, just to give you an idea. So it's a really relevant unit today, okay, for everybody. Because wherever you're, just for you to know if you're not going to a medical field, and if you're a medical field, people are going to ask you about it all the time. So all living things, seawater contain millions of viruses per milliliter. Pasteur didn't even know about viruses. He coined the term virus, Latin for poison, that rabies was caused by a living thing smaller than a bacteria. He didn't know about acellular and cellular forms, but he realized it was something he thought it was being transmitted, but was not something he could see under a microscope. Pretty clever. The real basis of studying virology came from the biggest cash crop in the early years of this country: tobacco, tobacco, tobacco. They fought, if you're, depending on how good you were with your history, if you remember the Spanish Armada and Queen Elizabeth, what did they fight that over? Tobacco. The Spanish wanted to get those tobacco plants because it was a cash cow. Okay, be clear about that. So Ivanovsky and Bergerac, okay, showed that a disease was called tobacco mosaic virus. It's a filamentous virus. Leffler and Frosch discovered an animal virus that caused foot and mouth disease in cattle. Again, these were crops or forms that were basis of the economy. So when fluids from host organisms passed through certain kinds of filters, the filtrate remained infection. That proved that the infection would be caused by a fluid containing agents smaller than bacteria. So are they alive? What are their evolutionary roles? What are their characteristics? How can they be so small, simple, and seemingly insignificant to be causing disease and death? And then understand, I'll say this number again and again, 15 to 30% of all cancers are directly related to viral infections. And the one that probably accounts for half of those, HPV, human papillomavirus, and genital cancers. Be very aware of that. So understand there's stuff here that's there. To me, I consider them non-living. I don't think there's a debate. They're unable to multiply, and the only way they can replicate is hijacking the host cell's mechanism. You remember the animation about transduction. They invaded a bacterial cell, they fragmented the bacterial's DNA, which is its control mechanism, and took over. Okay? So infectious particles, infectious molecules, whatever you want to call them. Even though viruses do not exhibit most of the life processes, they can direct them. So they're not certainly inert or lifeless. So I agree, active or inactive. But at least a good thing for the most part about most viruses when they're on surfaces, you know, many of them just degrade. Okay? That does not include certain kinds of what we will learn are called naked viruses, viruses that do not have an envelope. And the naked virus or the non enveloped virus, like the aforementioned HPV, like the common cold, like this one that one of my colleagues was talking about in the school district, norovirus, used to be called Norwalk virus, are naked viruses. They don't have an envelope. They last for a long time on surfaces. And we get those problems with them more commonly. At least you'll be happy to know that COVID and influenza and HIV are enveloped viruses. So a good disinfectant will take care of them. It will. Good news. Hey, I got to give you something positive today, don't I? Trying to get your attention. It's not working. You still have post Super Bowl. Terrible. Okay. So, do they infect cells and influence their genetic makeup? Absolutely. Did they shape some degree of evolution? Probably. 8% of the human genome, us, consists of sequences that come from viruses. 10 to 20% of bacterial DNA from viruses. So, obligate intracellular parasite. It's not just viruses. There are certain bacteria that are the same criteria. They are obligate intracellular parasites. Lyme disease, the most common in the United States of the vector-borne diseases brought to you by another species, in this case, a tick. Okay, is by definition an obligate intracellular parasite. It affects us as it comes from the bloodstream and the cells from the tick, and then infects us when the tick lights on us and seeks a blood meal, okay, which they do. So they cannot multiply unless they invade a specific host. And we'll get to the specificity. Again, that's referred to as the range. The range is not geographic. The range is the number of species that it infects. So influenza infects a lot of species. Rabies infects basically all mammals. Has a tremendously wide range. Measles, us. Okay. I am thankful that my dog will not get the measles. I'd hate to have to take care of him. I couldn't keep him in bed all day. Just forget it. You can't get chicken pox either. You can get into trouble. His problem is he's a snacker, and his particularly the kitty litter. If you get my drift. Ugh. But I digress. Okay, instructs the genetic and metabolic machinery to make and release new viruses. Look, I'm feeling old. My son was 40 on Sunday, my oldest, and none of you are 40. You sure? My daughter will be 37 in 2 weeks. So, obligate intracellular parasites of bacteria, protozoa, fungi, algae, plants, and animal. That's a big number. Like if 1 trillion is 10 to the 12th, that's a large number of viruses. 10 times the number of bacteria and archaea combined. Ubiquitous means they're everywhere. Major impact on biological life, ultramicroscopic. Not cells, very compact and economical. At a minimum, genetic material, you heard me say this 100 times, either DNA or RNA, never both. That's the number one reason to me why they're not living and they're not made out of cells. Okay. And a protein shell around them, famously called a capsid. Okay. So they independently do not fulfill the characteristics of DNA or RNA, never both. And now you get some real interesting oddballs, which we don't deal with in this aspect of virology. I mean, it's an interesting story to see how I have to teach our classes today, yourselves included, based on different than the way I was. I was taught virology in, don't smile, by a doctor named Jay Satz. His last name was Satz. He was the lead virologist in the Commonwealth of Pennsylvania. I will share with you the anecdote about what happened during Legionnaire's disease when we get to the respiratory diseases. Okay. He got up in the classroom, in a big lecture hall, like amphitheater style, and he kind of drew this. And he said, that's pretty much what we know about a virus. Actually, he was drawing the rabies virus. And it's got a shell of protein, and this is not a common look for a virus, by the way. And it has some kind of genetic material inside. That was how he started the lecture. So think what's happened over the last 50 years with regard to our understanding of viruses over time. And this is before molecular biology. This was before domains like prokarya and eukarya. We just had kingdoms. We had plants and animals. So it's changed dramatically. And most of that is molecular biology oriented when we began to look at the ultrastructure of things that were there. So you have different types. You have double-stranded DNA, single-stranded DNA, which you don't normally see, single-stranded RNA like we have, double-stranded RNA we don't have. Molecules on virus surfaces, that's the specificity. The name that we give these molecules are typically attachment or spike proteins. That's what allows them to attack whatever the range is. So COVID had, as far as we know, it's respiratorily spread. Okay. So it's got spikes on it that attach to the respiratory lining cells. Those wonderful pseudostratified ciliated columnar epithelia I'm so famously fond of talking about. Okay. So theoretically, if you got them on your hands, it's not a problem. Okay. But the reality is, and I say this all the time, 5 to 8,000 times a day we touch our face. And so either you're going to inhale, maybe there's some impact with swallowing. Obviously, because we're not 100% sure of how it's transmitted, though we think it's respiratorily spread. And what if you have an open cut or a sore? How does that play into it? Because those are all little nuances of these things. So when we look at the way these are spread, we're going to identify them as the most likely or the accepted ways that they are spread, as opposed to could they be spread some other way, which they could. So those attachments are very, very specific for the most part. Multiplied by taking control, that's the obligate intracellular parasite. And in some way, shape, or form, like if you remember looking at the animation for transduction, what did it do? It made tons and tons of little virus particles inside that bacterial cell. And if you didn't get a chance to see it, go back and take a look at that animation. It's on that list, and I have to post that list. I'm kind of like doing housekeeping this afternoon, like doing PowerPoint housekeeping and you know, Blackboard housekeeping this afternoon. It's on my list, my chore list today. And what's going to happen is by putting all those viruses in there, they reach a certain size, we call it burst size, and they pop out. That's one way. Interestingly, it's not how COVID does it. Okay. COVID takes over, influenza takes over. Okay. And what it does is it fills that cell and changes the cell into a little factory. And it kind of spits out. But while the factory stays intact, it spits out. We call it budding or shedding or extruding. There's a lot of different names for it, but basically it spits out intact. And you'll see some very good micrographs demonstrating that for your viewing pleasure. So they don't have metabolic enzymes. Okay, some of them come with certain enzymes. HIV is a prime example of that. And basically they have to take over the protein synthesis, all the intracellular machinery. So there are different ways. For a long time, we basically had two categories. Phage, which we talked about earlier with transduction that affect bacteria, which is short for bacteriophage. They're unusual. They infect us indirectly versus animal viruses. They're modifying that because we found out it was more than animals. It's other members of eukarya as well. Host and disease, structure, chemical composition, similarities. So there's a lot of taxonomy. Thankfully, you won't have to know it. So if I say enteroviruses, there's a couple of members you have to know. If I say rhino means nose virus, okay, basically it's a common cold. So there's some major, and we'll have a list of all of these and the members that you'll have to know. Some whatever the representative examples. When we say it's interesting, a category that's really important called paramyxoviruses. Okay, has 4 players in it that are important. Has measles, mumps, RSV, which is a big time player right now, real problem, and something called para-influenza, which is kind of like less than powerful flu-like. It's alongside. Para means alongside. So some of them have more than one category. So we'll look at the categories specifically. We'll look at whether or not they're DNA or RNA viruses, and there's a big distinction. Okay. You're going to see in general DNA viruses like HPV have an alarming tendency to become latent and to incorporate and alter host cells. We can read that as cancer. And again, HPV is poster child for that. Because once it becomes part of the cell, the DNA incorporates into the DNA. Whenever you have an alteration in DNA, we have a very famous name, which you just heard: mutation. And it's not the only player in this. Herpes viruses are in that player. And there's more than one herpes virus. But I'm being kind to you. You only have to know about 7. Don't thank me. Just have so much fun doing this. I don't get a lot of pleasure in life. We had a very good day yesterday. My stepson took over his grandma, and my wife and I got to spend Valentine's Day together. Goodness. Gee, I haven't seen you in a while. The dog and I and her, we had a group hug when she came in. Group hug. Obligate intracellular parasites. We'll discuss their size relative to other microorganisms. Here's a hint: they're smaller. The capsids will distinguish between envelope and naked viruses. Hold your eyes if you see a naked virus like this. We try not to have any inappropriate viruses here in the class. Okay. The importance of viral surface proteins or spikes, just told you a bit about those. We are not going to diagram the possible nucleic acid configurations that viruses possess. They are small. So if you think of how bacteria are to us, you've been in the lab. You're using oil, immersion microscopy, right? You're magnifying it 1,000 times. Is it easy to see an E. coli? I don't think so. Okay, they're small. E. coli are probably to us as viruses are to E. coli. So where most of the viruses, thousands of them can fit inside of an E. coli. It gives you an idea of how small they are. Okay, the largest virus, or one of them, there's several. Pox viruses are even larger. The smallest one, parvovirus, around 20 nanometers in diameter. Very, very tiny. Okay, where herpes simplex, fairly good size, 150. Okay, there are even some larger ones that are there. So let's say a red blood cell. I mean, a bacteria is probably 1,000 times more than that. Let's say easy. Typical E. coli, let's say, probably around 1,000 times more than that. Who has problems with parvovirus? Say it. Dogs. If they ever had the word sick as a dog, it was from parvovirus. When my first wife and I were married, we had Springer Spaniels. Okay, 2 of them. They were uncle and niece. Just saying. Reggie and Trudy. And we boarded him because we went away. When we came back, he got parvo. And I mean, vomiting and diarrhea of blood. Awful. Interestingly enough, you get parvovirus. So there's a strain of parvo that you get. We'll get to that. And it's relatively common. Okay. Relatively common. Thankfully, it's innocuous. And interestingly, it's not anything life-threatening or things like that. So there's a whole lot. I mean, when you see, and it's interesting when you hear about with your animals and things like that, and you go to the veterinarian, they have a lot of good information about this stuff that are there. So, I mean, it's a good idea to take a look at some of these things. There's an E. coli. Down there, yellow fever virus, polio virus. Look at the size of that. Here's a yeast. Yeast cells, maybe, and you're probably in the lab, did you do some yeast where you looked at it under high power and stain it maybe with methylene blue? Is it ringing a bell? You did a thing called a wet mount? You didn't do a wet mount. Okay, sorry. Just shut up, dude. Nobody cares. Okay? But that's one of the things you can see those with high dry, which is 400, as opposed to having to use oil. So you can see some of these that are there. This is what they look like. They're very, very small. So we have to use electron microscopy, and we glean something from these forms, whether they're in a more, and you're going to see these are called helical forms, and these have a fancy name. We call them generically isometric because all the sides are the same, but it's not really their true name, but we'll get it. They bear no resemblance to cells, lack any of the protein synthesizing machinery in the cells. Okay. Viral structure is composed of regular repeat units. So the capsid has identical protein units that form in a particular shape. They're called capsimeres. And you'll see when we get to that one that I said was isometric because everything looked the same, it's really got surrounded by a series of equilateral triangles, okay, that each have 20 of those capsimeres. And what it is called an icosahedron. You'll see. An icosahedron is like a 20-sided. If you ever have a game with a 20-sided die, most games you don't have 6, sometimes 8, but you get some very interesting games that have 12 or 20-sided dies. No? I give up. Have you seen him die with more than 6 sides? Thank goodness. I mean, I even think Feed the Woozle has more than that. Can't beat a good woozle. Okay, so regular repeat units, only those parts needed to invade. It has a coat, DNA or RNA, sometimes a couple of enzymes. The capsid, the shell. Collectively, when we look at the capsid and the genetic material inside, we call it nucleocapsid. Naked viruses, that's all they have, plus the spikes that protrude. The envelope is a modified piece of membrane, typically from one of our cells or a bacterial cell. And the spikes are on the envelope, on the membrane. So they protect the nucleocapsid and allow the virus to dock with host cells. So the fully formed virus, in a name that you often will use, is called a virion. The big distinction, and I'll leave it because we're just about out of time, between a naked and the idea is that people would think, well, it's got an envelope, it should be harder to kill. That's not, and again, I use the term kill, harder to destroy. The reality is the envelope, which is made out of fat, is a very useful target for a lot of different solvents, even alcohol, because it interrupts membranes. The spikes that it attaches are on the membrane. If you do away the membrane, it can't attach. It's useless. Whereas naked viruses don't have that rather easy, accessible target. That's why they are so much more difficult. Wiping something down with alcohol before you give an injection has absolutely no impact on naked viruses from a destructive standpoint. It might mechanically, you might move them away, but soap and water would do that. Water would do that. Rubbing would do that. Good enough for today. Thank you. We'll stop.